Amino acids are the fundamental building block to all proteins. So proteins are just composed of long chains of amino acids, and the type of protein or the function of the protein is entirely determined by this order and length of this polypeptide chain. So I'm going to talk about today the basic structure of every single amino acid and how they come together to form a chain of amino acids, and then I'm going to talk briefly about the three different categories that we put these amino acids in. So right at the top here, you'll notice that I've drawn a R group. So this is the side chain, and this is actually what determines which amino acid it is. There are 20 different amino acids, and they fall into three subcategories, and the R chain is entirely what decides what amino acid it is and what properties the amino acid has, and thus what properties the protein has. Because if this amino acid is incorporated in a certain section of a protein, the properties that this amino acid have determine the properties of what the protein as a whole have. And then you have this connected to what is called the alpha carbon. So this alpha carbon right here, this is the alpha carbon. It's called the alpha carbon because it holds together the three different groups here. It is the carbon that it is in the center. So this is alpha. And on the left here you have what is called the amino group. So this is an amino group because this is an amine. It is a nitrogen that is attached to a carbon and then three hydrogens. So if you had a long polypeptide chain that ended with this amino group, it would be called the N-terminus. And on the other side here we have a carboxyl group. So a carboxyl group is just a carbon with a double bonded oxygen and then a single bonded oxygen on the other side. And if it ended in the polypeptide chain ended with this, it would be called the C-terminus. So here we're going to look at how two of these molecules, two of these amino acids might come together to form two amino acids here. So right here we have one and we have two. This side right here is your amino group right here. I'll circle it for you to make it a little simpler. So, in green we have our amino groups right here. Then in pink we have our side chains, the R's, like this. And on the other side in orange we have our carboxyl groups. All right. So we can see here that I actually omitted by accident a carbon here, so I'll draw that in really quickly. There is another carbon that this is bonded to, so this would be an H. Forget this O, this is just a single bonded H, much like this side over here. And then you have a carbon right here with your double bonded oxygen right there. So sorry about that. That is an H right there. So right here you would have a condensation reaction between this OH group and this H group. So right here, this will come down to form this, and you will have an H2O that leaves and goes off somewhere else. And then you have a peptide bond, which is a covalent bond. So right here, this is called a covalent peptide bond between amino acids. And then you have two different R groups. So these R groups, I'm going to talk really briefly right now about there's 20 different amino acids, and I'm going to talk about the different groups that, or the categories that they get put into. So they're grouped according to the properties of their side chains. So the first group that we have is called nonpolar side chains, which are hydrophobic. Nonpolar. And then you would have a polar side chains, which are hydrophilic, meaning they love water. So these ones hate water. Hydrophobic just means water hating. And then the third group is electrically charged side chains that are hydrophilic, so polar charged. Once again, they love water. And then I like to also show that there are three unique side chain properties for three different amino acids. So I will talk about them right now. There is glycine. So glycine is just super, super small. So it can be tiny, sorry, it is tiny, and it can be either hydrophobic or hydrophilic. So I can draw this structure. Actually, no, I'll do that in another video. I'll leave it at that. So let's just put tiny, polar, or nonpolar. And then we have cysteine, which has the ability to form covalent bonds with another cysteine, which form a disulfide bond. And last but not least, we have proline. And proline will form a disruptive kink in a polypeptide. So I'm actually going to briefly read off the list just so you have an idea of the names of these amino acids, and I encourage you to Google a picture. There's lots of charts that have all of the amino acid structures with the different R chains or side chains that are attached to it, and you can see what each one of these look like. But the first section that we have, the non-polar side chains, you have glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline. And then in your polar side chains here, you have serine, threonine, cysteine, tyrosine, asparagine, and glutamine. And then for your electrically polar charged hydrophilic side chains, you have aspartic acid, glutamic acid, so those are your negatively charged ones. And then you have your basic, which are positively charged, which are lysine, arginine, and histidine.